Hi everyone. Um, so I'll have a few different videos on metastasis, which is the topic of this video. Let me put that at the top. Metastasis. So I'll have a few different videos that step through how this process works and the different steps, with just like a quick video for each step of the process. But yeah, we'll jump right in to kind of the start of metastasis and how this works. So metastasis again is going to be when cancer cells spread. So we have a primary tumor right here. This is our primary tumor. And metastasis is when these cells break off. They enter the bloodstream. So they'll enter the bloodstream, travel along to a new area, exit the bloodstream, and then reestablish in a new area. And this is our second tumor in the body. Right, so that's what metastasis is. So let's jump right into how this process happens and what it's going to look like. So step one of this process is going to be a process called cell detachment. And really, this is going to involve a couple of different changes in terms of the molecules that are expressed by this cell, what those look like. So if I have my cell here, I'm going to kind of write out what's normal in a healthy cell over on the left here, and then we'll talk about the transition that happens for a cancer cell. So a normal cell is going to have kind of these traits that are called epithelial. And that largely refers to kind of a collection of these traits in terms of proteins that are expressed, as well as this trait of being kind of low mobility. So we'll put that as one of our traits, low mobility. Some of the other traits are going to be presence of a couple different proteins. First, we're going to see the presence of a protein called E-adherin, which is going to be present on the surface of the cell. And then secondly, we have another protein called beta-catenin. So to kind of walk through what these two proteins do, E-cadherin is responsible for connecting cells together and allowing them to communicate. So how this might look in practice is maybe I have another E-cadherin molecule over here. This would allow me to interact with another cell over here. Two cells. This is my other cell right here. So these two cells would be able to interact using these E-cadherin molecules right here. Right, so that's kind of our example of what we're looking at in terms of E-cadherin. And there would also kind of be some signals that could be sent either direction via this connection. Okay, And then secondly, beta-catenin anchors this molecule, or sorry, E-cadherin anchors the molecule beta-catenin to the cell membrane. So beta-catenin in pink here is tied to the cell membrane by E-cadherin. And beta-catenin actually operates as a transcription factor, which means that its job is to kind of facilitate the transcription of proteins in the nucleus. So when beta-catenin is tied to the cell membrane, that means that it can't interact in the nucleus with any genes, and it can't transcribe anything. So this kind of deprives beta-catenin of its ability to do anything by anchoring it to the membrane. This is our normal epithelial cell with these two proteins interacting in our kind of low mobility state. Now if I transition over what cancer is going to look like, I'll have cancer over here. Here's my cell. Let me put my nucleus in for good measure. So that is going to be my nucleus right here. My cancer cell is going to have a little bit of a different characteristic. This is going to be called a mesenchymal kind of characteristic that this cancer cell has. And this is when it is looking to metastasize in particular. So this trait, it is specific to metastasis, right? So cancer cells will transition away from epithelial to mesenchymal when they're going to metastasize. And it has a couple different traits that are different as compared to what we saw in our epithelial cell. So the first is going to be that it has increased mobility as compared to our epithelial cell. The second is that it's going to have a different connective molecule. So there's going to be a decrease in E-cadherin production, and instead we're going to make something called N-cadherin. Right, and N-cadherin is going to be responsible for allowing different mesenchymal cells to communicate. So E-cadherin was specific to those epithelial cells. N-cadherin is going to allow messages to be sent back and forth in communication between these mesenchymal cells. So this is connection and communication again. Right? So very similar to E-cadherin, just going to have a slightly different purpose. Right? And then lastly, if we look at our beta-catenin molecule, it's not going to be associated with our N-cadherin. Instead, it will be present in the nucleus. If I draw kind of my quick little arrow over here, this is my beta-catenin, my transcription factor in the nucleus. This is going to be transcribing things now, so it has its activity back, transcribing. And in particular, it's going to be transcribing pro-tumor proteins. So proteins that are helpful to the tumor, right? Okay, so let me zoom back out. So our step one, just to kind of review and think about what this is going to look like, is that cancer cells are going to make this transition from this more epithelial nature over to this mesenchymal nature. That's really going to be the essence of cell detachment. We see an exchange of E-cadherin for N-cadherin. We also see an exchange of beta-catenin for being tied to the nucleus, and now instead it's been, or tied to the membrane, it's now able to move into the nucleus. And then lastly, we see this change in mobility, where our epithelial state, we have low mobility, whereas cancer cells, if they want to metastasize, they need high mobility. So they're going to transition into a high mobility state. All right, so that is an overview of this first step of metastasis. In the next video, we will talk about our next step, which is that basement detachment idea.